All right, I think everything's working. Can everyone see my screen that I'm sharing?
Yes.
Sweet. And you can hear me okay?
Yep.
All right, well, we are just going to get started then. See if I can get the slides to change. There we go. All right, so Catherine has passed the torch to me to provide an introduction of the Ecosystem Monitoring Program. And I am also going to talk about the Action Effectiveness Monitoring Program. And I will give a brief overview of the data collection that happened in 2022. Then we're going to talk about a new idea of changing how we report out and share data with the Ecosystem Monitoring Program. Ian's going to give a quick overview of Tableau and how that software is going to help us essentially reimagine the reporting in the Ecosystem Monitoring Program world. And then kind of a timeline for these steps of transitioning the data and reporting. All right, so for the benefit of those who are not aware, and probably maybe folks who see this online, because I know everybody in the room is fairly familiar with our Ecosystem Monitoring Program and our Action Effectiveness Monitoring Program. These two programs really work hand in hand. The Ecosystem Monitoring Program, its main goals are to track the status and trends of the ecosystem conditions in the Lower Columbia River Estuary, focused on the sort of ideal or reference condition tidal emergent wetland habitats that are so critical for salmonids. And these data help us evaluate restoration success in our Action Effectiveness Monitoring Program, which focuses on monitoring restoration projects that post-restoration. So we use this data together to help guide, design, and update restoration practices. And we're going to talk a little bit more about that in this intro discussion. However, today's presentations from the researchers are focused on that Ecosystem Monitoring Program data. So the Ecosystem Monitoring Program has been around, started in 2005, and the way that this data has been collected across the estuary has changed over time. However, the goals and objectives have stayed the same, primarily focusing on looking at spatial and temporal variability across the estuary, focused on salmonid habitat and food web dynamics. And again, really focused in these tidally influenced emergent wetland habitats that are so critical for these species. Now, this is mainly funded through BPA, and we work with a lot of researchers to collect these data. And I'm going to talk about those researchers as we move forward in the talk. So as of today, we have 5 major sites that we monitor across the estuary, and these are stratified across the 8 hydrogeomorphic reaches. Now, a hydrogeomorphic reach in the Lower Columbia River estuary is essentially it's been categorized as having unique hydrology, unique ecology, and so we like to stratify our sampling across these so that when we have restoration sites within these, we can compare the conditions, because within each reach you have a slightly different conditions that you'll observe. Now, as I mentioned, the way that we have sampled and the sites that we have sampled in the Ecosystem Monitoring Program have adapted and changed over time. But right now, since 2012, we have primarily focused on these 5 major sites. So Owako Slough is at the mouth of the estuary in Reach A. Then we have Welch Island in Reach B, White's Island in Reach C, Campbell Slough in Reach F, and then Franz Lake in Reach H. So these 5 sites receive the full suite of ecosystem monitoring, which is the habitat conditions, abiotic, water quality conditions, food web dynamics, fish occurrence and abundance, macroinvertebrate occurrence. And so I'm going to talk a little bit more about those as well, but these 5 sites receive the major monitoring. And then we have 2 additional sites, the Cunningham Lake, which is in Reach F as well. Cunningham Lake is a habitat site, so we monitor all the habitat conditions there. And this is due to the fact that Campbell Slough has issues with grazing. Now, in the habitat talk today, Sneha is going to talk a little bit about our approach to working around grazing in Campbell Slough, but Cunningham Lake is essentially a really good site that does not receive grazing as much as Campbell Slough. And we have a lot of reference sites in Sauvie Island where Cunningham Lake, restoration sites in that area. So we use Cunningham Lake data for that restoration comparisons as well. So that's why we monitor Cunningham Lake and Campbell Slough and Reach F for habitat conditions. And an additional site that we consider part of the ecosystem monitoring program is Steamboat Slough, which is a restoration site that is part of our long-term biomass detrital flux study. All right, so that's kind of a quick overview of all the sites that everyone's going to talk about today. And who is everybody that does this research? Well, we have a really amazing team of researchers, and this is not a history of who have done this work. If you would like to know more about all of the different researchers who have contributed to this research over time, it's been many folks. We have all of that information in the ecosystem monitoring report, and happy to share that with you. Today, Joe Needoba from OHSU is going to talk about the research he leads on Mainstem, water quality, and site abiotic conditions. Here at the Estuary Partnership, we lead the habitat structure, hydrology, soil, sediment accretion dynamics, and detritus monitoring. Sneha is going to talk about that today. Today, Tawnya Peterson from OHSU is going to talk about food web dynamics and isotope analysis that she does for that to inform that work. Fish, prey, and macroinvertebrate analysis is led by Jeff Cordell, Jason Toft, and Kerry Accola. Kerry is going to be talking about that research today. And then, of course, we have fish, community, and occurrence data collection. That was historically led by Reagan McNatt, but she has now moved on, and we have Curtis Roegner, who is going to be doing that work. Susan Hinton and Jeff Grote, I believe, are going to present today. Or is it just you, Jeff?
Just me today. Just me today.
Yeah. Jeff is leaving the charge.
Yep.
Cool. And then we have critical field support by April Silva and Ryan Elsmar. They essentially help with all the data collection, including, outside of the scope of habitat. They also help OHSU and UW with their data collection. All right, so real quick, this encompasses all of the data collection for the ecosystem monitoring program. Now I'm going to talk about the action effectiveness monitoring program. Now, earlier I mentioned that these two programs work hand in hand, and so I did want to mention this. The action effectiveness monitoring does have a different schedule for monitoring. So for ecosystem monitoring program, we do all of this all year long for all the sites every year. For the action effectiveness monitoring, it's rotated across the different restoration projects depending on their age. And so it's a little bit more complex, and I'm not going to get into that exactly, but I just wanted to mention that we do collect the same suite of habitat conditions across these sites for level 2 sites, which receive a full suite of habitat data. And we also do macroinvertebrate community data collection. And at years 5 and 10, we do fish community and currents. And this is important because this synergy between the two data sets allows us to do that evaluation analysis of restoration progress over time. And another thing that this allowed us to do is because the action effectiveness monitoring program has a different sort of data collection approach. It's a little bit less data for each site than all the ecosystem monitoring projects. We were able to transition this report this year into a hybrid format where we have taken a lot of that data and analysis and moved it online into data dashboards using Tableau software. Ian's going to talk a little bit more about what Tableau software is, so I'm not going to get into that right now, but I just want to talk about how we just published this in August, and you're welcome to explore it and see how we have reported out on these sites. In these reports and dashboards, we've included a lot of ecosystem monitoring program data as it is essentially correlates with those different sites we are reporting out on in the action effectiveness program. Now, why would we want to hybridize a report? Well, for the action effectiveness monitoring, it makes a lot of sense in my mind that we would want to shift the data into a place where everyone can interact with it and look at it. Project sponsors can have a little more access to those reports and the essentially evaluating how their sites are doing compared to reference sites. Well, for the ecosystem monitoring program, I feel like there's even a larger need to transition this report. Currently, it's about 400 pages long. It takes us months, between us and the researchers, it takes us months out so many hours to update this giant report that is essentially a textbook. Because it is a warehouse for all of the data and reporting since the program started, it is quite untenable for a layman or really anyone who is not a technical expert to really dive in there and extract information from it. So it's time for a change. And with Tableau, we can push a lot of that work online and make it really a lot easier for folks to interact with the data and even take the data and use it for themselves and do synthesis work with it. So we're really excited for this transition, and we have a little bit of a timeline that we're going to talk about today. So I'm going to talk about this timeline, and then I'm going to hand it over to Ian, and he's going to talk more about what Tableau is. Between Ian and I've got our Tableau wizard hats on. We are really excited for this transition. So this year, we are going to start working with the Ecosystem Monitoring Program researchers to incorporate Tableau into their workflow and help sort of think about how that data needs to be structured for it to go into Tableau, and how we can essentially automate some of the long-term stats and trends analyses and table creation so that takes that off their plate and makes it a lot simpler for them and gives them more time for really important synthesis and manuscript work. Part of this is going to be unpacking the ecosystem monitoring report. We're going to take the methods and protocols out of the report and have them in a standalone document. I've been talking about this, creating a standalone protocols document or an update to the protocols for a long time. We're about at an 80% right now in that draft, and we're going to be shopping it out to the Ecosystem Monitoring Program researchers soon for them to edit it. So this is going to be a standalone document we can essentially link into a new and improved hybrid EMP report. Then we have the programmatic history, which is currently included in the the EMP report. We'll be pulling that out as well. So again, we're unpacking the report, making it more of an executive summary, analysis, synthesis document that is very approachable. And then if you would like to look closely at data, you can online. We feel like we're going to be able to do a fully hybridized report in 2023 and 2024. This year, we'll do a partially hybridized report. All right, so I'm sorry if that was a little bit dry, but I wanted to kind of unpack that a little bit for everyone, and so everyone's on the same page. Now I'm going to pass it to Ian, and he's going to talk about Tableau.
Well, so as Sarah has said, the only thing we really have to live for is Tableau, as in Princess Bride. And I'll be talking about the use of Tableau for reporting and data sharing. So, next slide, please. Thank you. So we really need a proper data management system. If you want like the full details of this, visit our April Science Work Group chat. But with the EMP data, we have so much data, and it is so complex, and it's like time series data. So we have like drone stuff, hydrology, biomass, isotope stuff. We have just a lot of very complicated data that is mostly in time series format. So it really excels with Tableau. So Tableau can and will really help us synthesize all of this data. Next slide, please. So what exactly is Tableau? So Tableau is what the cool kids are doing now. It's a data visualization software with advanced analytics capabilities. It's mostly used by the finance sector. However, it's rapidly being adopted by the scientific communities. It emphasizes seeing and understanding data insights for both the analyst and the audience. It's kind of like the next step in the world of visualization. And the next slide, please. So we all like pretty graphs, I hope. So reimagining the EMP report. Our vision is basically similar to the 2022 AMA report, which is a two-part reporting style. So there's the traditional section as well as the Tableau section. Next slide, please. So Tableau really excels with like that kind of multi-part format because it allows you to like embed different documents into the Tableau. So the methods and background would remain in separate traditional documents like Sarah mentioned earlier. There would be standalone dashboards with the results and discussion, and a brief executive summary level written report. And then ideally there would be links from both the report and the dashboard pointing at each other so you can kind of toggle between everything. Next slide, please. So Tableau particularly excels with time series data and geolocated data. So all of the EMP data, to my knowledge, is basically time series data as we're looking at long-term status and trends. So it really is phenomenal for aggregating those time series and comparing them across different statistics and all of that. So these are the hydrographs for Ruby Lake and Cunningham Lake. which are two action, or Abiotic is an action effectiveness monitoring sites, and it was very quick and simple to calculate. Next slide, please. So Tableau is also really good with looking at semi-related data to analyze them together. So in this case, we have sea level rise and net accretion. The only thing related between these two data sets are the river kilometer of the site and the fact that both of them are rates of change. And that's it, but we can still plot them together and compare them directly. And next slide, please. Tableau is also really good at creating summary statistics and indices from the raw data quite quickly and complicatedly. So these graphs summarize the relative vegetation of Ruby Lake and Cunningham Lake over the years. And you do this once and you never have to do it again, basically. You just update the database. It's all good. Tableau also emphasizes self-exploration and accessibility. So in this case, we have a diptera highlighted on this. And if you don't know what a diptera is, you would click on anywhere where it says diptera and it would change that picture in the slide. And it really helps people who do not know their macroinvertebrates to understand what every single macroinvertebrate is. And then next slide, please. So what are our next steps? Ideally, we will be transitioning all of the EMP datasets into Tableau, either this year or next. We'll do a full data synthesis analysis at that same time. But after it's all incorporated into Tableau, of course. We're aiming to include the drone as quickly as possible and complete a full synthesis analysis of all the sites. We're hoping to answer critical uncertainties at both the site level and across the lower river. We're updating the monitoring protocols and methods and finalizing the standalone programmatic history document. If you think your data is too gumpy for Tableau, you haven't seen the drone stuff yet, but so it's doable for everybody, I hope. Next slide, please. If you want to look at any of these examples online, here's a bunch of links for you. The Action Effectiveness Monitoring Report page is probably the best one to go to. It's specifically look at the release key dashboard, and yeah, contact us with any questions. Next slide. Thank you.
Oh, do we have another slide?
We have one more, I think. Thank you, everybody who has helped collect data and funds us.
Thanks, Ian. Does anyone have questions? Catherine, do you have a question?
I was just gonna say, is Tableau your side gig? Is that you guys are getting money from Tableau? Is that right? From all those memes? Or, yeah.
We haven't
They
figured
really
that one
love
out yet,
Tableau.
Catherine. You're keeping us too busy to have side gigs. I see a hand. Is it Jason?
Hi, that's me. That was really interesting. Thank you for giving that overview. I haven't used Tableau much at all yet, so there's a lot. It's exciting. It'll be really fun to learn more. And I have a nerdy question that's maybe too nerdy for discussion of everyone, but you know, we use R for all of our analysis, and R is of course powerful and free and all that. How can that integrate with Tableau? Is there a way for the code to transfer over and all that?
You can directly integrate all R code and Python code into Tableau, and it's pretty seamless if you know how to do it.
That's awesome. I'm excited to learn how we can do that. So, all right, thank you.
I think Ken.
Yeah, hi, thanks, Ian and Sarah. That was great. It sometimes it's hard to hear what you're saying because I was trying to read your funny cartoon pictures and laughing to myself. But so thank you for putting all that together. And maybe you said this, and I'm sorry if I missed it because I was looking at the cartoons. But like, where does the data sit? Is it in your own like SQL database? Or I guess I'm trying to understand. You know, I get the tools in Tableau, but just where's your data, or did you have to migrate all your data into a new sort of database?
That's a great question. So most of our data is kind of stored in Excel documents still and like CSV files, but Tableau does something where it creates an extract, which is it automatically creates kind of a Postgre database, and then it just pulls directly from that for everything. So it's a lot faster and more seamless than the CSVs.
So you're just a follow-up question is, so you have Excel spreadsheets that are on a server and you're just pointing to those, you're sort of mapping that data using Tableau so that you can use the tools to summarize that data?
Yeah, basically.
It does connect to other data sources. And we have as you can see with the ecosystem monitoring program, the complexity and breadth of data is vast, and then the drone data. So we are working on coming up with other sort of data warehouse solutions that Tableau connects to. It's something that we're doing a lot of research on right now.
Cool. Thank you. Yeah, it's really interesting. And I think it'd be interesting just to track that over time and hear some of your solutions, especially, like you said, with the drone data that takes up so much memory and space.
We'll keep you posted. I think Nick had his hand up first.
I did, yes. I was waiting for a Lord of the Rings reference for one ring to rule them all. How did you learn about Tableau? Like, how did you decide that this was the platform that you wanted to use? And an add-on question, is this being used elsewhere in the industry?
The answer to that question is because of COVID, I was tracking the data through the Oregon Health Authority really closely, and I was like, what is this interface they're using? It's amazing. And it was Tableau, and I had been told by others that I should check it out before that. So I checked out Tableau, and I started using it a couple years ago now, since like in 2020. And it's really amazing. We found it really amazing for our use. And then we've seen other researchers out of UW using it for climate change analysis and data. So it really is catching on in the science industry. If you want to learn more, Nick, we will share a link to our April science work group meeting where all I do is talk about how I figured out that Tableau seemed like an okay idea. Kerry, did you have a question?
Yes, thank you.
Yeah, that was a great talk, great memes also. My question is, so if I'm a user and I'm online working with the data, do I have to pull data from multiple sources, or can I combine them like you said, even if they're semi-matched data, and pull it into like one CSV? How does that work?
That was a lot. There are a lot of different ways. I think the answer is, and I'm just going to answer it in, is there's just like a lot of different ways to combine data. And, one thing that we're not necessarily convinced we need to do is create one place to hold all the data to rule them all. We think that potentially one way of doing it is having the researchers individually manage their data with and report out with Tableau. And then as that happens, we can kind of culminate a central warehouse of data. But as long as you have something that ties the data together, so you do have to have like a data structure, like the site name or something. You could tie it to that, even if it was a different metric. If that is also your question.
Okay. Yeah,
maybe
I didn't ask it clear. It's a Tableau question, which is like I see that you can plot things that may not be fully matched, but can you pull data to combine into a file with Tableau, or maybe just yes? Okay. Thanks.
All right. Does anybody else have questions? I feel bad. I don't want to take up too much time from the rest of the talks today, but I appreciate people are interested. Catherine, you're muted.
I was going to say we have plenty of time at the end too to ask more questions if people want to follow up. We could move over to Joe's talk if folks are done. Okay. I think you're on the hook next, Joe, right? Talk about Mainstem.
Yeah, I'm here. I'm here.
Okay.
I'm sharing my screen, I guess, right?
Yep. The little up arrow, right?
How's that look?
I can see it.
Okay, good morning, everybody. My name is Joe Needoba. I'm associate professor in the Environmental Systems and Human Health track of the OHSU PSU School of Public Health, which is a relatively new school. I'm going to be talking today about abiotic conditions. I'm really going to just try to make a couple simple points, given that we have a lot of data, and I'm looking forward to learning Tableau along with everybody else to help better visualize our long-term complicated data sets. I think it's worth just sort of talking a little bit about where I am, because it's not familiar to most of you. This is the new Vanport building of the OHSU PSU School of Public Health. It's right in the center of the Portland State University campus. Just opened in March, and the School of Public Health occupies the 5th and 6th floors of this building, and we share it with City of Portland and a few other entities that occupy this really beautiful space. Maybe one day we can have a science work group meeting here. It's a nice central location, and it's a new building, so it's exciting. And it's, I just wanted to sort of, and this is a sort of a bit of a sidetrack, but it's called the Vanport building. And from a School of Public Health perspective, there's a public health angle to the Vanport flooding of 1948. If you're not familiar, this was a big change to the city of Portland when this area flooded and basically wiped out this neighborhood and forced all these people to move. Many of them were low income people that were working in Oregon and were displaced. And so the Vanport building, I think, was named in part to recognize that important aspect of public health. But for the purposes of my talk today, it's also interesting that this event occurred because it was a particularly wet year with a large mountain snowpack followed by heavy rain in May, which led to the Columbia River overflowing the levees and flooding this area. So by May 25th, 1948, both the Columbia and Willamette rivers reached 23 feet, 8 feet above flood stage. So that was a historic event. One of the reasons why the Columbia River Treaty with Canada was implemented, it was one of the policy arguments for establishing the Columbia River Treaty, where 2/3 of the water stored in the dam system is held in Canada. I just like showing this picture of Mica Dam because I used to go there as a teenager. There's good fishing back behind Mica Dam and Kim Basket Lake. So it's a long time ago, but I thought I'd bring it up. And what all of that has done is changed the nature of the flow in the Columbia, as most people here know. And the spring freshet has decreased in its peak discharge, and therefore we have less chance of flooding now, and we wouldn't see a recurrence of that Vanport flooding very likely because of the dams. And also the peak discharge has shifted to earlier in the year, and that's a little bit of a climate change story, I would believe. This is from a thesis published a few years ago on modeling historic Columbia River flood impacts. And so I just wanted to explore our data set with that in mind. And I've showed these graphs before, but if you haven't seen this, I just explain really quickly. These are different years of the Columbia River discharge at the site about river mile 50. And what we're seeing here is on any given day of the year, from January through to December, there's a maximum flow, and that's what I'm plotting here. I'm taking all the data from 1997 to 2022 and plotting it as a maximum on any given day and a minimum and an average, and then overlaid on that is the actual year. So in 1997, you can see we had a relatively large freshet, whereas in 2001, if you look at the upper right graph, there really was no freshet whatsoever. In fact, all of the data from 2001 falls on the minimum flow rates for the data set. So that was clearly an extraordinarily dry year where we didn't see a lot of flow. So a lot of variability occurs in the, and it really is important for the ecosystem monitoring program because the flow of the river really impacts our marsh sites. And so you see in 2005, and then in 2011, another large flow year with the freshet making up much of what we know is the maximum flows seen during this whole period. So I like to characterize these years as being these high flow years or low flow years or normal average years. 2015 was another low flow year, and 2017 was a very high flow year. So the variability in the flows, as many people know, is incredible. So what's been happening lately? So here's the most recent few years. And what we see is that 2019 and 2020 were basically normal average years. And then 2021 was an incredible drought year. Most of you will remember. The way that I can think back about these is which years were good spring Little League baseball seasons. And so 2021 was an amazing Little League baseball season because it didn't rain. But in 2022, we basically got washed out. So this year was a wet year relative to 2021. So with that context, then I wanted to just talk about some of the main stem conditions. And I'm just going to focus on temperature because that's what most people really are interested in. And here you can see our three big flow years in this data set, 97, 2011, and 2017. And on the right is again the maximum temperature on any given day in the data set and the minimum. And then in blue is the temperature of that year. And so in all three data sets, most of the time the temperature is cooler than average. In 2011, it was one of the coldest that we've seen the river through the spring and summer. And this is important from a fish habitat perspective where temperatures don't get too warm too early in the season when fish are using that habitat. And then in our low flow years, 2001, 2015, and 2021, last year, we see typically much more warmer conditions in the spring, and 2015 being the year that stands out as the warmest of the data set. And I've trying to think this year, and maybe with the help of Tableau, putting this together in more of a predictive approach and saying, well, if we know what controls the flow and the discharge, we can sort of understand better what the marsh habitats are going to experience because they typically follow the temperature of the freshet, or sorry, of the main stem, and if anything are warmer than the main stem. And so I think the question is, well, if 2001 was such a low flow, lower than 2015, why does 2015 have the warmer temperatures and 2021 as well? And really, I think the short answer is it depends on the snowpack, which contributes to the flow. And then it depends on local air temperature conditions. And really 2015 was a really warm spring and led to warmer conditions than we saw in 2001, where the spring conditions were actually, we didn't have a lot of warm days back then in 2001. And so we didn't see these really warm conditions. So to put that into some context, here's a figure of the data for the number of days where we saw temperatures above 19 degrees Celsius for each year. On average, it was about 57 days. And this is just from May through August, and from the year range of 1995 to 2022. And I think that in summary of this, I say that in 1997, we had a high snowpack and a warm summer. And in 2011, we had a a high snowpack and a cold summer. And you can see the impacts on temperature. We really stood out in 2011 as being one of the coolest of the main stem temperatures. And then in 2001 and 2015, we saw low snowpack and a cold summer. And then when conditions aligned, we had a low snowpack and a hot summer, leading to certainly the most warm days that we've seen. In terms of how the standard deviations from the mean, certainly 2015 stood out, as it did, you'll remember, with ocean conditions that year, and we had the North Pacific temperature anomaly. So that was my sort of attempt to summarize how I think the importance of the river for controlling these basic conditions, and how it relates to our EMP sites. So I just want to go over them quickly. I find that these products from the Estuary Partnership are really nice to think about where our sites are relative to the rest of the potential ecosystem marsh. So the green is existing priority habitat. The yellow is technically recoverable habitat, and the red is lost to development and paved over or what have you. And our sites in this area of the estuary, on the left is Owako and Welch Island on the center here, give you a sense of where they are. And this is what they look like. On the left is Owako in the early spring, when the marsh hasn't really grown, it hasn't begun to grow yet. And so I'm showing this because this is the methods that we're using to collect the water quality data here. These pipes. This is the USGS approach, where your instrument is at the lower end of the pipe, but you can access it from the other end. When the tide is up high, you can still get access to your instrument by pulling it up out of the pipe. The central picture is Welch Island. You can see our pipe there. And there's a picture of NOAA taking us out to the sites, going fishing near Kathlamet. In the Reach Sea, our site is there. That's White's Island. And this is what it looks like. You can see our pipe is on the left there, mostly underwater. We since extended it further up to get more access to it. In Reach F is Campbell Slough. As Sarah mentioned, this site has some grazing problems, but it's a long-term data set where we have a lot of nice data, and it really has a large change in its hydrology. On the left, you can see a freshet where most everything's underwater. And then the same area later on in the summer, you can see that the channel is much shallower. And here's a picture of our instruments that we measured water quality with, is the YSI XO2 sonde, and just doing a little bit of calibration with the cows in the background. We don't have any EMP sites in Reach G, and then our final site in Reach H is tucked in there, and that's Franz Lake Wildlife Refuge. And this is an interesting site because of the beaver dam that typically is built there, and you can see our pipe. So I don't want to use up the whole day talking about our data, so I just wanted to summarize a couple of things that I think are important and related to the earlier part of my talk. Well, here's one more method slide where you can see us installing those pipes. Those are students that have worked in the lab in the past. And the YSI instruments are becoming more effective at capturing multiple parameters. And so we're currently measuring pH, chlorophyll, phycocyanin, so getting a measure of phytoplankton as well as cyanobacteria, and then dissolved oxygen, temperature, conductivity, and that other sensor on the right is nitrate. And so YSI has started to produce an optical nitrate measurement that we're test driving in the estuary this year. So here's a summary of data since 2015 for temperature, and this is the number of days above 19 degrees Celsius and in the actual marshes. And so Owako, Welch, Whites, Campbell, Franz are shown as the columns, And you can see 2015 is the first set, and then all the way down to 2021 at the bottom. On the left is above 19 degrees. On the right is above 22 degrees. And the trends here follow what the Mainstem showed, although we didn't have a lot of data in 2015 for technical reasons. The data that we did have all showed nearly 100% of the days in the months of June, July, and August were above 19 degrees Celsius, whereas in other years like 2017, we didn't see really any days above 19 degrees Celsius for many of the sites until mid-July. And this is the type of pattern that I'm hoping that Tableau will help us to better communicate. I will say that I think there's a potential for improved models to fill in some of these temperature data gaps, and so looking forward to these types of relationships that will allow us to go back and maybe fill in some of when these dates, knowing the Mainstem conditions and the river flow. One more graph here of data. So one of the questions is, how are these sites eutrophic? Is there a lot of hypoxia? And the answer really is not as much as you might expect. And so places like Campbell, where it's relatively disconnected from the Mainstem, long residence times of water, and despite high productivity, I think that the nature of the channel and the shallowness of it doesn't lead to a lot of hypoxia. And so we're able to see over the last 3 or 4 years, we've had multiple different discharge conditions in the river, yet we don't see very many instances of oxygen going below 2 milligrams per liter. Bronze, because of the beaver dam, I think is more susceptible to that. And our report goes into this in a lot more detail than I can do today. So in conclusion, I think it's important to think about the snowpack and the spring and summer conditions as they relate to Mainstem temperatures. Of course, there's a lot of management associated with the dams that can help change that as well. I didn't talk about that today. High temperatures at the EMP sites reflect Mainstem and watershed conditions, and dissolved oxygen levels indicate few instances of hypoxia in the EMP sites, which is good. So I think moving forward, this long-term data set can support both statistical and numerical model development, and something that I'm interested in doing to help better understand the role of abiotic conditions in the EMP sites. So thanks for that. I hope I was fairly coherent this morning. Here's some nice pictures of all the field work that we do. I feel pretty privileged to get to go out on these, and so I have students that come. So shout out to Narayan and to Crest for taking us out on, and in the picture in the middle is some of our students doing sampling at the Kamis-Washougal dock, where we have a site. And on the left is an iPhone 14 Pro picture of White's EMP site at low tide.
And there's his sidekick selling iPhones. Any questions for Joe? Have a couple minutes. Kind of moving a little bit, but slow, and a little bit behind Jason.
Hi, I don't like uncomfortable silence, so not that there was one yet, but I'll ask a question. Maybe I missed this, and maybe I should know this already too, but that's why I ask questions. So is the 19 degrees benchmark that you showed a lot for being above that, is that more just a benchmark that you're like, okay, 19 degrees is a hot year, or is that related to some parameter, say, of juvenile salmon, response or fish response in any way?
What I've learned over the years is that 19 degrees is the number that is sort of used as being above the healthy threshold for juvenile salmon. And I'm sure it's rooted in some science. Maybe Curtis could speak to that, or someone else can speak to it, but that's the number that gets passed around. as a benchmark for juvenile salmon habitat?
Yeah, excuse me. Yeah, exactly. 19 is a benchmark number. It's been used based on a lot of lab studies and things. 16 is determined to be like the optimal temperature for the juveniles coming down. Things below that are okay until you get too cold, and then their metabolism suffers for that too. So there's like an optimal range that have been determined, and definitely the 19 degrees is what is said to be bad. Now, having said that, you know, the fish can live definitely at those higher temperatures. They just need to eat a lot. There has to be a lot of food to keep their energy balance going, because they'll metabolize at the higher temperatures. Without that food, they starve. So, and they can grow really fast, apparently, at the high temperatures if they have enough food. So it's this benchmark, but you can't really say that it causes mortality necessarily. Same thing with the oxygen, though. You know, you did show a bunch of days, Joe, where the DO is around, I thought, 6 milligrams, and that actually is been shown for fish to show behavioral changes in their position. Like, they don't like it like that, and they'll move around to try to find higher oxygen concentrations. So, you know, with the oxygen thing, there's kind of a behavioral threshold as well as a lethal mortality threshold, kind of like the temperature thing. So both those things kind of work together too. So the thing is, the fish don't like it with too low, and they don't like it necessarily too hot. So maybe the combination of those things is kind of a good framework to think about, like, where would the fish be?
That's all really interesting. Thanks, guys. And like, it just seems maybe with the future of Tableau, I mean, there's definitely an opportunity to link everything you just said to what the fish are eating. And, you know, we have the diets over the years. That would be really fun to look at.
Totally. Yeah. One other thing I just throw out there for the fish part is that, you know, there's all these different stocks in the river, and they migrate at different times. So a lot of the fish that are of interest for people, upriver fish, a lot of those ones are already gone before the temperatures actually reach any kind of potential lethal problem. But there are definitely groups that are, you know, stay the whole year in some of these areas, and they actually might even be genetically predisposed to withstand the higher temperatures. Something we haven't really looked into, but there are definitely stock-specific migration patterns that we see in the genetics and things that can kind of feed into all this as well. So a lot of caveats with, you know, with the fish and where they are and when.
Cool. Thanks, everybody. Any other questions for Joe, or are we moving on? Oh. Okay, I think our next presentation is Sneha and Sarah discussing our habitat structure. And we might kind of move things around a little bit. We're supposed to have a break around 10:30, so we're going to try to stick to that break at 10:30, if that's okay. So we might just move Tanya's presentation to after the break, if everybody's okay with that. Groovy. Sneha, are you ready?
One second, I'm trying to move my presentation over to like the next window so that it becomes more seamless and, you know, all the stuff.
And I'll just say that Sneha is going to take it away. She's on this presentation today, so thank you, Sneha. I'm just here for moral support.
Do we have lots of memes? We have lots of memes, Sneha. I mean, yeah.
No, Ian and Sarah are the meme kings and queens. I'm more of the let's talk about pretty pictures.
Okay, so many pretty pictures.
All right, can everyone see my screen okay?
Yep, looks good, thanks.
So hey everyone, it's great to see all of you all again. Today I will be talking a little bit about the EMP habitat data that we are predominantly going to use in some of the focus studies that we're currently working on. Now, Sarah did an amazing job explaining the EMP and the AMER programs. I'm just going to touch a little bit on the habitat objectives. We monitor certain ecological components, and that is we look at different inundation patterns at our long-term trend sites and how these vary over different years. We also collect vegetation composition data. and evaluate their variations over the years as well. We also study sediment dynamics across these sites, and we collect plant above ground biomass samples to be able to talk about the primary productivity, and this feeds into the salmon food web dynamics of the program. This year, we collected the habitat metrics across all 5 of the trend sites and Cunningham Lake, which is in Reach F. We've also collected above ground biomass data at Steamboat Slough. Now, every year we run these stats, but this year I thought I would share this with the larger group. We collected about 900 plots of plant community data, and we collected like nearly some 400 bags of plant samples for biomass analysis. Each of our team crew members also hiked like nearly about 55 miles trying to collect all of these data. So I mean, I have to say it's a pretty good workout for people who are like tracking steps and, you know, are all into the step challenge and everything. I have to say, I definitely win those step challenges during the summer. Now, moving on to the main crux of the presentation today. Ian did an amazing job talking about how Tableau can be used for different data management and analysis. Since we've started extensively using Tableau, we've also been able to crosswalk our large data sets from the habitat. And as a result, our team has been working towards synthesizing these data into manuscripts, some of which I'll be talking about today. Now, our first publication, well, this has actually been published online, and this is something that Sarah and I originally spoke about in 2020, and then Sarah and Ian were talking about it in 2021, as well as in, and we showed the published dashboard in the June 2022 Science Work Group. Essentially, the goal of this approach is to integrate multiple aspects of wetland recovery, including native and non-native plant community development and changes in salmon habitat opportunity into a site-wide assessment of ecological condition. In addition to monitoring hydrologic conditions over the site, wetland plant composition data determines not only the habitat quality, but also the amount of organic material that goes into the juvenile salmonid food web. While our traditional approaches are like we collect good data with it, but we are only able to capture about like 5% of the vegetation and topographic changes. On the other hand, using UAVs allows us to capture ongoing vegetation and topographic changes across the entire site. So by combining these two methods, we are able to measure the existing habitat conditions, as well as talk about the existing dominant native and non-native plant communities, salmon habitat access, as well as opportunity, and it helps us to predict any impacts from climate change that can occur. We applied this approach at one of our action effectiveness sites up at Waluski, Astoria, and we were able to essentially demonstrate the salmon habitat quality across the entire 200-acre site. And as you can see, we were able to show that in April of 2021, salmon were able to access 124 acres of native wetland habitat. Now, the goal of our study is to be able to essentially emulate this model across all of our long-term status and trend sites. Now, the next publication that we'll be working on is going to be looking at the ecological drivers of reed canary grass, as well as other native and non-native vegetation species across the lower Columbia River estuary. From our EMP research and other researchers in the region, we have been able to confirm that site hydrology is a critical driver for reed canary grass abundance. We have also been able to show through our EMP data that marsh elevations receiving less than 30% indentation, whether this is through tidally dominated or fluvial sites, that's like a threshold for reed canary grass dominance. Along with these varying indentation patterns and marsh elevations, they also influence the soil characteristics and create the biogeochemical template that's required to establish wetland plant communities. In our study, we will be exploring these relationships and mechanisms further and talking about how soil chemistry influences reed canary grass and other native vegetation species in our reference as well as restoration sites. The next publication that we are planning to work on is related to sediment dynamics across our reference and restoration sites. We have data from 2008 that we collected across the EMP and other AMER sites, and we plan on basically talking about what trends we've seen and what sort of and essentially, like, one of the big things that we've seen is that the marsh elevation actually can influence some of the accretion and erosion dynamics that we've seen. Predominantly, that the lower marsh accretes a lot more than the high marsh. We also plan on studying the net erosion and variability in the data that we've seen at some of our sites, which we attribute to grazing and trampling. And in order to study this, we actually installed more sediment stakes in Campbell's Slough on the other side of the channel, which we've seen to not be as grazed as much. Another aspect of this study will be understanding how our tidal wetlands are keeping track with sea level rise. For this study, we used the US Army Corps' adaptive hydraulics model, which provides sea level rise in meters across 50, 75, and 100 years. We compared the net accretion and erosion rates from our EMP sites to these scenarios, and we noticed that Franz Lake currently is the only EMP site that is keeping up with the forecasted sea level rise. We also anticipate White's Island and Cunningham Lake to keep up with these scenarios. Continued monitoring of these metrics will essentially provide us critical information for future restoration and management objectives so that we can combat or at least try to mitigate some of the wetland loss that can occur because of sea level rise. Now, the last study that I will be talking about today looks at the net above ground primary productivity, which is essentially about the salmon food web. The goal of this approach is to evaluate species-specific biomass and nutrient contributions to the salmonid food web of the Columbia River Estuary, as well as explore the mechanisms driving these contributions. We changed our sampling protocols in 2017 to include detritus sampling, and this allowed us to understand not only how much of biomass changes to detritus, but also how much of the organic material either accumulates or is then exported out of the site. Through our long-term EMP sites, the sites that we collect biomass are at Welch, White's, Steamboat Slough, and Franz Lake. Through our study, we were able to observe that the percentage of biomass density at each of these sites would change based on the year as well as the season. In Welch Island, during the summer at high marsh zones, we've seen biomass density be dominated by Lingbii sedge and reed canary grass. whereas when wintertime approaches, we've seen an increase in biomass of forget-me-nots or like Mysotis laxa. In the low marsh of Welch Island, we've seen summer biomass be predominantly dominated by spike rush. Similar trends we saw in White's Island as well, where the high marsh was dominated by reed canary grass and lingvii sedge, and the low marsh was again lingvii sedge and spike rush. Moving over to Steamboat Slough, during the summertime, we noticed that the high marsh was dominated by reed canary grass as well as soft rush or Juncus effusus, whereas the low marsh has been completely dominated by Wapato. And over at our more fluvial Franz Lake sampling site, we have seen a 40% reed canary grass to a 60% water smartweed or Polygonum amphibium split in the high marsh, whereas the low marsh biomass density is dominated by Wapato. Now, we look at our biomass composition through seasonality, which means through summer and winter. Looking at the seasonality, we also observed that the largest flux or change in standing biomass happens during the summer and winter time period, where some of this standing biomass then gets converted to detritus and eventually then goes out of the site. The largest flux of detritus out of the wetland occurs during the spring and the summer period. And we observed that between the summertime and the following spring, we see a gradual increase in the detritus material, whereas between the spring and the summer, we've seen a sharp drop. We also noticed that the low marsh biomass abundance is very high during the summer. However, during the winter time, most of this above ground biomass has already converted into detritus and been exported out of the site. And our winter biomass is predominantly dominated by the high marsh species because of their dormant stems and their stolons. In 2020 and 2021, we saw these as highly productive years. And we anticipate this is because of the longer growing season, and especially for Franz Lake, there used to be a beaver dam that got like blown out because of the storm events, which meant that there's essentially the site was not holding as much water anymore. So we had there's a lot more low marsh area for the water to essentially dominate. Now, what is the importance of like learning about primary productivity? And the essential thing is that the marsh plants provide a lot of higher quality, so is a higher quality source of energy when compared to phytoplankton and algae for the macros. So studying all of this and the nutrient composition as well as the amount of biomass provided by the native plants in low marsh and high marsh is critical to understanding the larger food web of the salmon habitat in the Lower Columbia Estuary. Also, learning about all of these mechanisms would help us understand how these may change in the face of climate change. Well, now that I presented all of these studies, we are continuing to synthesize our data to look for like more trends and hopefully create crosswalks between like the data that UW, OHSU, NOAA, they all collect, so that we can talk about like how each of these merge and like talk more about the food web dynamics. We also are interested in studying about how the sediment dynamics as well as seasonal river discharges influence the primary productivity at the site. Lastly, I'd like to thank all of our crew members that make this data collection possible. The Habitat program collects a large sleuth of data, and this would not be like we wouldn't be able to do this job without our fellow researchers over at CREST, Narayan, April, They are awesome in the field, and like we've enjoyed their jokes and all of that. We also had some really great field interns this year who helped us process all of the biomass samples that got collected. And of course, our colleagues over at Short and Associates who help us with the plant community data. Thank you everyone for listening. And yeah, I'll open the floor up for any questions if you all may have. Thanks.
Great, thank you, Sneha. We have some time for questions.
Wow, did I explain it so well that I don't have questions?
That was a lot of information. Oh, Jason.
Hi, it's me again. This is all just so interesting. I have a question about the plant community because in our lab here in Seattle, we've been looking more at plant community too around Puget Sound, and we've been trying to get more direct insect associations by using a sweep net and so characterizing vegetation at sites and do direct sweep net sampling within those vegetation types, which gives a different signature than say the Newston toes, right? Or a fallout trap, which is more indicative of a larger area. So I don't know, it's a rambling thought of future work, but I'm just wondering, Sneha, if you have any thoughts on like, for being out in the field so much, you know, on what questions or next steps might remain with linking your data to future data collections.
That's a good question. And like we've been trying to integrate the macro data to answer essentially these sorts of questions. Well, when we're in the field, we have seen that based on like the water years, we've seen like the plant community and as well as the macros communities change. So whether these two are kind of interrelated or whether there is something else that's going on that's, you know, driving these changes is also something that we're interested in looking at. We also want to know how these interactions essentially change the food web dynamics, or like how the isotopes would change based on all of these interactions. So yeah, there are quite a few questions that we can answer.
I'm going to add to this that a couple hot takes is that, you know, we're in the field and we definitely are bombarded by bugs. So, you know, we have our own anecdotal assumptions about year to year macro communities. You know, some years are pretty Diptera heavy and sometimes they're the biting kind. I think it would be really interesting to see if we could pick up any of those signals comparing the data sets. One thing that we've noticed in the AIMER data where we have tried to synergize the data is that in the history of the AIMER program, for whatever reason, we have chosen many different methods of monitoring macroinvertebrates instead of staying with one method. And so when you have many different methods of collecting macro data, it becomes less comparable over time. And so I think that sticking with a method, which is luckily with the EMP data, I know that we have very consistent data. So I think that's going to be easier for us to unpack that story, hopefully, once we get that EMP data online.
Cool, thank you.
I think Ken, Petra, you had a question? And then
Yeah,
Curtis.
thank you. Yeah, thank you, Sneha. And I really like the photos on one of the slides that were sort of like icons for like the predominant vegetation. So thanks for using that. Once it was kind of overwhelming at first, but as you walked us through it, it was helpful to kind of understand what the predominant vegetation type was. So I really like the way you did that. I had a question that's very different than Joe's. I noted you were showing sea level sediment accretion and predicted sea level rise.
Yeah.
And I was wondering if there's been this interest in carbon storage. Are you guys, and I didn't see anything presented, but do you guys have an interest in understanding, you know, like, so carbon and you guys are doing biomass, so that potentially could be a way to look at carbon storage, and just curious, because that's kind of a, blue carbon, and there's the state of Oregon's interested in how, our natural lands can help mitigate impact from climate change.
Yeah, that's a great question, Ken. While in the EMP program, we have studied the nutrient deposition, like we have studied nutrients in terms of like plant biomass. The carbon storage in the sediment, we have touched on it a little bit, but we will be touching a lot on it moving forward next spring, because we have another program where we're going to be monitoring carbon dioxide and methane that's coming out of some of these fluvial and intertidal wetlands. So yeah, once we have those results, it'll be pretty interesting. Like, we're all very excited to start that research for sure.
Yeah.
We're going to be working with Joe Needoba and Tawnya Peterson from OHSU to get that study going. So yeah, we're very excited. Yeah.
Thanks. Well, I'll be excited to see what you guys learn in the future. Thank you.
Yeah, Ken, it was like that. That was like we planted you, because I was wanting Sneha to talk about that carbon and methane flux study. And so thank you for allowing us to give that a shout out.
Thanks, Ken.
I think we have 2 more questions. Curtis, Roegner?
Yeah, excuse me. Yeah, the fluxes I think are really interesting, and especially with the prey dynamics. And what we found in the lower river is that the tidal flux of insects out of a lot of these areas is quite significant. And I always wanted to kind of expand our AIMER study using of the prey flux up into some of these areas. So I'm just trying to plant that seed, perhaps, of perhaps a focused study on some of these other not as tidal areas to look at what's actually being produced from the wetlands that gets exported to the wider ecosystem. For the lower river, we're finding that it's quite significant. And when you add up what comes out of just some of these little streams that we study into the whole, like say, Willamette Bay, you can appreciate that these wetlands are producing a ton of food that's being available for some of the bigger fish, especially, that don't go into these small channels, and they're more regulated, deeper water. So just a little seed out there, perhaps, that I think would be super interesting to expand the flux study to some of these other areas.
Yeah, nice.
Great, thanks. Laura, this will be the last question, I think. So, and then we'll take a break.
I was just going to say, so I know, well, and Sarah, you were there with this too, but the work that Aaron Peck and Rob Wheatcroft did at Woluski, looking at carbon storage and carbon loss in 2018. And I was actually thinking that maybe it could be interesting to see some of their work presented during this, unless you all did this back in like 2018 and 2019, and I just wasn't part of the group. But yeah, I know that they looked at the amount of carbon that was lost when it was diked off, just anything that's built on since then. So it was less of a question, more of just like, all right, I guess the question is, have you all tied that work into this?
No. So we haven't, I mean, Sarah has told me that there has been carbon studies done in Woluski. Like she I think she put it as part of her dissertation. She did say something.
Laura and I were out there together collecting data with Aaron. So I think I was like, I think
on the sledgehammer a lot.
Yeah, she has a lot of information about like Woluski and like the number of studies done there. We do plan on integrating these over time, but currently we're going to start by monitoring the landscape scale ecosystem fluxes. First, we're going to start by putting up a few flux towers at a site and studying the landscape scale. And then over time, we're going to be putting in soil chambers and studying the soil cores. So yeah, it's a pretty big, like, long-term study. And so in the spring, we're going to be starting with the landscape scale ecosystem stuff first. But yeah, it would be pretty interesting to see how it changed.
I just want to segue with this is that essentially we are, Sneha and I are really well, and so is Catherine, really well aware of the work, the blue carbon group is doing, which is part of that Aaron Peck research. And we are trying to essentially create some synergistic energy and data for these more freshwater floodplain systems. And so we're starting like in Reach F, and because it's fairly well established data collection in the saltier regions, right? So we're starting up in these more freshwater systems that there really isn't a lot of good methane carbon flux data for. And then hopefully, I would love to put an eddy tower out at Woluski, but we got to start, we're starting in the fresh and we're going to move our way down to the salt, I think, maybe. What do you think, Sneha?
No, thanks. I forgot to mention where we're starting in, where we're studying. Yeah, correct. Yes, thanks, Sarah.
Thanks, everybody. So I'm gonna say we're supposed to have like about a 10-minute break. I'm gonna shorten it to 8 minutes. Is that okay? So my clock says 10:38. So how about we come back at 10:45? Does that sound good? Not quite 8 minutes. Okay, awesome. 10:45. Thanks, everybody.
I'm trying to see where I can share.
Oh, there's
Oh,
like
I see it. I see it. Yep, I see it. I see it. Yep. Trying to get used to Zoom, WebEx,
Yep.
Google Meet, and Teams, and got it.
It's a transition. Like none of them are really. They're all the same and different. It's like using a credit card machine.
Yeah, totally. Oh geez.
Are those plants on your shirt, Sarah? Is that?
Yeah, it's biomass.
Oh. Okay, my house is kind of cold. I might actually turn the heat on. I don't know.
You don't have that mini split right there. I can't remember how to turn the heat on with the mini split. I just know how to turn the AC.
Right? I think you have to go down to the mode. Oh wait, you have a different. Yeah.
Which one do you have?
I have Mitsubishi. You have to go. It's like a mode key that you hit. Sorry.
Yeah, I just have to like look at the manual.
If nothing else, maybe it'll clean up the air quality in my house too. Oh.
Yeah, do you have a replaceable filter on yours?
I just have to wash it every so often. Yeah, can replace it every so often too, but to wash it.
I think the
In
answer's
a
on
sink.
Tableau.
Whether I can, how to turn my mini split from AC to heat.
Yeah, right?
I'm sure you're right, Nick.
Instructions in Tableau, yeah.
I'll have to pick your brains. Mini splits are in my future.
I love
Schedule it now.
mine.
So they can start manufacturing them.
Yeah, we waited 8 months for ours to get installed. Supply chain issues are real.
Sign of the times. All right. Well, I see 10:47. I don't want to hold up the day too long. Should I just get going? I'll just jump into it.
Go for it, Tawnya. Thank you.
All right. Well, I'm working from the title that appeared on the schedule today. So it's a little bit different than in the past. And I'm trying to lean into the conditions affecting part of this. So that's where I'm going to kind of go, taking some data from this year, but also kind of contextualizing with information from the past as well. The work has been that's the team at OHSU, but also folks at the partnership, Estuary Partnership, students past and present, and others. So I share the acknowledgement with everybody about where this data come from and what we do with it. Okay, let's see if I can figure this out. I don't normally present on Teams very often. So we had some really nice talks about what's going on in the EMP. I just want to underscore that the part that we cover in my group or team is looking at the food web from the aquatic side. So I don't talk about the plants here, but it's, I think, implicit that the plants and invertebrates are part of all of this. And so I just show this, my version of the food web. Sneha just shared the really nice graphic that was produced for one of the synthesis report back, 5 or 6 years ago. But really, we're thinking about what goes into the fish in terms of the food web, the prey items, and then the primary producers that feed the invertebrate prey to understand really how the base of the food web is operating based on conditions with river flow and temperature and all of that. So we move up from biogeochemistry and abiotic conditions all the way up to what's getting into the fish. And our goal is to really understand what the fish experience and what they're able to eat. And that's where we sort of work together. As an overview, the conditions that affect the salmon food web include water quality. That's what the salmon experience. So we had some nice discussions among the group about the importance of dissolved oxygen and pH. That also affects the physiology of primary producers, which was also covered a little bit through what Sneha had to say. I'm thinking about the organic matter fueling prey. That's the primary producers, including plants and phytoplankton in the water. And we measure nutrient availability, or the nutrients that are there, temperature. Light is important. We have to consider physical processes like flushing, retention, mixing, flux and transport of materials. So that's kind of the last discussion that Curtis was talking about, so the insect prey fluxes. There's lots of things to think about in terms of the growth of all of the different levels, as well as what's coming in from elsewhere. And that's all available to the fish. And then, of course, competition and predation, which we don't talk about. The data that we collect, water quality, we have, as Joe was talking about, the continuous in situ sensors that measure things like nitrate and pH, temperature, conductivity, all of that. So that's what the salmon experience. Biomass, that includes chlorophyll, the species composition and abundance of phytoplankton and zooplankton, as well as the particulate organic carbon and nitrogen. So we're trying to fill in the gaps with respect to what's in the water, the organic matter, and the lower food web that use that organic matter or produce it. And then finally, food web and productivity indicators, and that's the isotopic signatures of carbon and nitrogen at multiple trophic levels feeding into the salmon food web. And we also look at the isotopes of nitrogen and oxygen in nitrate, and I'll talk about that just a little bit later. So we'll just start off with a little bit of information about water quality. So we've heard a little bit about 2021 as being a productive year for the plants. In the water, just kind of to remind you what Joe was talking about, there was very low river flow in 2021. And as a result, and I know you can't really read what's in these graphs, these are the 6 sites, the 6 EMP sites, Ilwaco, Welch, Whites, Campbell, sorry, 5 sites, Ilwaco, Welch, Whites, Campbell, and Franz Lake Slough, and then a Mainstem in the middle just to compare. What this is showing, I'll just talk you through it, is that levels of nitrate are high in the winter, and they tend to decline as aquatic primary producers draw down those nutrients and start to grow themselves. But because the river flow was very low this year, none of that, there was, let's see, normally there's a dilution of the growth of the primary producers, and that didn't happen. And so after the winter flows, there was low nutrients after that pretty much at all of the sites. So very low nutrients, like kind of close to zero, which is somewhat unusual for the time series. So nitrate was very low in the spring and summer, and generally is high in the winter time, as it usually is. You can see at Campbell's Slough, if you can kind of peek through, those numbers get very low after about May. So lessons that we know from the Mainstem about the relationship between river flow, nutrients, and light and temperature. This is just showing some combination plots we've made over the years, looking at binning the different time periods from late winter, early spring, when usually productivity starts to get going, into the spring when it's the highest, and then the summer when it declines again. And what this is showing just is that the spring is the time period when chlorophyll and phytoplankton biomass is the highest. It accounts for most of the productivity for the whole year. In the summertime, light is more available, and so in theory you would have more productivity, but nutrients decline, and so that's why we see this suppression in addition to grazing by zooplankton. So a lot goes on to make those numbers lower, but really the lion's share of aquatic productivity is found in the spring. And yeah, that's all I wanted to say about that. So if we look at the sites, that was data from the Mainstem. If we look at the individual EMP sites and try to get a handle on what they look like on average, this is just showing the 5 sites: Campbell, Franz, Ilwaco, Welch, and Whites. And these plots are showing that on average that you might get similar amounts of phytoplankton biomass at each of the sites, but they're episodic blooms or high biomass events, mainly at Campbell and Franz. This is where we see cyanobacteria blooms or even high amounts of other types of algae in the summer months, when you get kind of a separation from the main stem. So you can get that retention that leads to higher biomass building up. And those huge tails are very, very high. Typically, chlorophyll would be, on the less than 10 micrograms per liter, and in some cases at Franz, it gets up as high as 100 for short periods of time, but it can get quite high. The lowest is always at Ilwaco, closest to the river mouth. If we look at a time series at a couple of the sites here, just to show how 2021 looked, this is a time series from 2011 up to 2021. And there's a couple years where you have higher algal biomass, 2014 and 17. 2021, way over to the right-hand side, looks pretty normal. It was a little higher than 2020, and that's because there wasn't that dilution by high river flow during that year. Looking at Franz Lake Slough, you get, so if you look at the y-axis is expanded here, so that actually goes all the way up to 100. There's some very high algal biomass sometimes at Franz Lake Slough, and that's happens when the beaver dam separates the exchange flow and things like that. There's some physical processes there that lead to this situation. 2021 was sort of in line with other years at Franz, and then at Welch, we're kind of zooming in. This is the chlorophyll doesn't tend to get as high, but 2021 again looks sort of like 2018 and 2014, 16. So it's kind of in line with what's normally seen. The type of phytoplankton that's there, we can combine all the data that we collect on phytoplankton species composition. And this is just showing canonical correspondence analysis, where we're combining environmental variables like maximum discharge, temperature, year of observation, and then some of the nutrients like phosphorus and orthophosphate and nitrate. And this just shows what type species are connected to what environmental characteristics. Every year we see that diatoms, the glass-shelled primary producers that tend to be the biggest ones that bloom right away in the spring. That happens as discharge is increasing. When nutrient levels are high, light tends to be kind of low. They're the ones that take off first. And then you see a succession toward other species like smaller, less nutritious really species like chlorophytes that include chlorophytes, cryptophytes, and chrysophytes. And so you can relate them to where the different environmental conditions. We've seen that there is a relationship, for example, between cyanobacteria prevalence and temperature, which is seen elsewhere, but also with phosphate concentrations. So we can do a little bit of predictive guesswork, sort of getting back to what Joe was saying this morning about trying to use what we know to then make some predictions about how things might be in the future. So we, as Joe was mentioning, we have in situ sensors in the water that measure biological components like chlorophyll and phycocyanin. Chlorophyll is the algal pigment that all algae have, and phycocyanin can be found in cyanobacteria. So this is a way to get a handle on water quality associated with the presence of nuisance species like cyanobacteria that can sometimes make toxins and cause problems. So this is showing a couple of years of data from the continuous sensor at Campbell Slough and Welch Island. So Campbell usually has, Campbell and Franz both often have cyanobacteria blooms or high concentrations in the summer months. So this is showing 2020, where there was a little bit towards the end of the year, winter gap. And then in 2021, there was pretty high abundances of cyanobacteria after the sort of into June. And at Welch, that's not seen as much. They were present in 2021, but not as high as at Campbell. But we can track this on a day-to-day basis, which is quite nice because they're a little bit elusive sometimes because they tend to float at the surface and we don't always see them. So Franz Lake Slough in 2021, like Campbell, had some high concentrations of that blue-green pigment into the summer months. So in general, over the time series, the years we've been looking at this from 2011 to 2021, we consistently see that early in the season, you have these nice big diatoms, then they shift to smaller diatoms or different species. And then by the summer and into the fall, you get a shift to greens and cyanobacteria. So you go from bigger species to smaller ones and presumably less nutritious over the course of that period. And so I just pulled out a reference to a study that was published in 2020 that showed that the assimilation of organic carbon and nitrogen from different phytoplankton species does make a difference for the primary consumers or the secondary producers. So in 3 different species or groups, so there's an amphipod, a polychaete, and a clam. In each case, this black bar is one species and the gray is a different one and the white is a different one. As you go across, the first part of this first graph just shows the percentage of the assemblage they were fed as being 100% diatoms and then smaller percentages. In each case, more carbon on the left and more nitrogen on the right were assimilated by the invertebrates when diatoms were available. So just kind of just making the point, I think it's something that I've mentioned before, but maybe not had the pulled out the data to show. But diatoms of the phytoplankton that are available are far more nutritious than cyanobacteria, cryptophytes, chlorophytes that are smaller and just have less lipid material in them. So the species make a difference for the invertebrates, and the invertebrates make a difference for the salmon. So there's a direct connection there. So we investigate this sort of thing by looking also at isotopes. Sarah made some reference to this. This is just a scatter plot of all of the different data points that we have from the time period between 2014 and 2021. And you can look at these data points in terms of their carbon and nitrogen isotopic signature and use the differences in the different groups to then predict what's making it into a higher trophic level, like salmon. So you have kind of characteristic signatures of each different type of group, whether it be vascular plants versus phytoplankton versus sediments and soils, whether it's terrestrial woody debris or whatnot. So we can use that in a food web model to try to predict the diet content. And I've done a little bit of this. It's still a work in progress where we're I'm really interested in looking at what the invertebrate prey are eating in terms of their organic carbon. So that's, thinking about the detritus that's coming into the river versus the in situ growth by phytoplankton. You can kind of distinguish those things. So looking at the chironomid diet and the amphipods, and then that can feed into the salmon. So I'm not going to really talk about this, but thus far, we can see some familiar patterns like the chironomids and amphipods, as we know from the diet data, make up an important part of the salmon food web. But we have to understand some things first before we can really do that. And so what I'm showing here is this is a time series of phytoplankton organic carbon associated with the phytoplankton over this is a couple of year period, July, January. So each of these peaks in gray is the spring bloom. And of those, this is the component of the spring bloom of phytoplankton that is large. So these are the ones that are making up the most nutritious part of the phytoplankton available to the invertebrates and to the salmon. And if we look at the isotopic signatures of those, they change from the beginning of the phytoplankton bloom to the latter parts. So as you get species succession or physiological changes or whatnot, they change to get heavier, both in terms of carbon and nitrogen. Now, heavier just means that there's more of the heavier isotope compared to the lighter one. This is how we look at the fingerprint of the organic material. And so this is important because it feeds into our assumptions about the food web model. If we were assuming that a particular food source or a source of organic matter is one number, when we can see that this changes, we need to adjust our thinking and our assumptions. And so digging into that a little bit. So they get heavier over the course of a bloom. And in part, that reflects the material that they are taking in. So we also, in addition to the particulate organic matter and the plants and the invertebrates and the salmon tissue, we look at the isotopes of nitrates, the very bottom of the beginning of the food web at the biogeochemical level. What's happening with the nitrate, the nitrogen and the oxygen in the nitrate molecule. And if you plot the month of the year versus the N15, the heavier isotope of nitrate, it increases over time. And this could be occurring for a few different reasons, but it could be suggesting that, or it goes along with the fact that there's a negative correlation with the concentration of available nitrate, and so could say something about the nitrate assimilation and productivity of that site. So we take that into account when we look at what ends up being assimilated into the particulate organic matter. And so when you look at the off-channel sites from Campbell Slough, Franz, Welch, Whites, and then the average of all of those together, I've left Owako out because it's a little different being more marine, and compare that to the Mainstem, the nitrate signature is heavier. And what we can interpret that to be is potentially an indicator that more nitrate is assimilated into particulate organic matter at those sites i.e. they are more productive than the main stem, which is consistent with a kind of a hypothesis that off-channel sites are more productive. This has been seen in estuaries elsewhere. It's not surprising, but this is another way to look at that and to try to get a handle on the sort of the rates of productivity without actually measuring the rates of productivity. And we also have oxygen isotopes that can help us try to distinguish the sources of the nitrate that's coming in. And so that's kind of a work in progress as well, but I think it's an interesting indicator. And just a couple more things to add. We talked about pH and dissolved oxygen before, and I guess what I wanted to say about this was just that when you have off-channel sites that are highly productive and maybe not as well flushed, this is where you can start to see fluctuations in some of the characteristics like pH and dissolved oxygen. So when you look at an hourly version of pH and dissolved oxygen at Campbell and Franz Lake Slough, which tend to be the places where you see the most biomass buildup, they fluctuate quite a lot on an hourly basis, with pH exceeding sort of this benchmark range of good quality habitat between 7 and 8.5 occasionally. And similarly with oxygen, you get little dips down to very low percent saturation relative to the atmosphere and very high peaks. So these are very dynamic systems, and that dynamism can impact what the lower food web is doing as well. If you average that though on a daily basis, which is what's shown here, for the most part, the dissolved oxygen is pretty good, and the pH becomes within that range of acceptable water quality. At least at Franz Lake Slough. At Campbell, we have some exceedance of that early in the season. So some of the questions that I'm kind of looking at with respect to these data are the off-channel habitats, what are the indicators that they may be more productive than the main stem? We can use isotope data from the nitrate to look at that, as well as the patterns in biomass accumulation and assimilation, as is also shown in the isotopes. And the continuous data support that. And just to revisit the conditions affecting the salmonid food web, nitrate nutrient availability is important. Nitrate was low in 2021. I didn't mention this, but phosphorus was about average. So really that nitrate was a pretty interesting signal. You need sufficient retention time for the biomass to build up. And when you have a small freshet like we did, that leads to lower rates of dilution, which leads to sort of the perception that there's more biomass around. But it's really a combination of a lack of dilution and in situ growth. And the pH for the most part was in adequate range. And that is all I wanted to say.
Great, thanks. Couple minutes for questions.
See if I can stop sharing now. I don't know how to do that. Oh no.
It's the same button you hit before, so it's up at the top next to the leave button, I think.
I hit it, but then it's not doing anything. I'll figure this out.
Any questions for Tawnya? There's probably a lot of information. Ken.
Yeah, thanks, Tawnya. I guess that last part I was just thinking that kind of dawned on me was the lower years, sort of less volume of water, right? So it's not only just sort of maybe less productive, but just overall your greater pool of food is lower too. And are you guys quantifying that in anything?
Yeah, you mean the abundance, yeah, we do that as well. So the chlorophyll, the quantity in micrograms per liter is one measure of that, but we also do the abundance of all the different species, so we can kind of compare those two things together. Sometimes the amount of chlorophyll per cell or per species can be different, so there's a little bit of wiggle room there. But yeah, we end up collecting all of that data just to have a look at the year-to-year variation. One part of that's a little more difficult to get at is the fact that some of the species are different sizes. So everything about carbon content, you can get differences as well in terms of whether you have one big diatom is the same as 87 small cyanobacteria. So trying to get a handle on that. Sometimes the abundance doesn't always tell you about the amount of organic matter that's in the river.
Thank you.
Great, thanks. Jason, I'm gonna you have a question, and then we're gonna move over to Kerry.
Got it. I'll be quick, I think. I'm in course, that was great, Tawnya. I'm of course intrigued by the amphipod and chironomid slide that you said you weren't going to talk about much. But I'm going to pay attention to Kerry's talk, so I'll set that aside. But, you know, when I'm thinking about that and differences in the estuary and the energy ration that we have for those, I want to steer my question to your earlier slides about diatoms and cyanobacteria to make sure I understand it, because it's more outside of my wheelhouse. And my question is, was that diatom signature consistent across the estuary, or were there locations where that fluctuated depending on where you are?
You mean the CCA, like where they, or which, like, what about the diatoms?
Well, maybe I just don't remember understanding it. Like, depending on where you are, what reach, right? If you're in a more freshwater or a more estuarine reach, is that a consistent signature?
Oh, that they bloom first and then they change to other things. Yeah, well, so it's really driven by the river, so the elevation of the river. So the winter will kind of bring in all the nutrients, and then you'll get growth, and then usually the water level's pretty high, and there's like not a lot of light and lots of nutrients, and you get Asterianella formosa will start first, this big colonial diatom, and then there'll be a succession to other species that are a little bit different, and then the freshet comes through, wipes them all out, and then you start again. And in that second phase of things, that's when you get either more lightly silicified species, and then also the greens and chrysophytes come then. So it's kind of a winter group, early spring group, and then kind of a post-freshet group. And that it's seen everywhere except El Waco. Waco's totally different.
Okay, cool. That was kind of my
thank you
for that learning moment. That was thank you.
Yeah, El Waco is almost hard to compare to anywhere else because it is so distinct from the other sites.
Interesting. Thank you.
Great, thanks. Thanks, Jason. Kerry, are you ready?
I am. Am I sharing?
You are. Perfect. Thank you.
And how is my sound? Sometimes it fades in and out. It's good?
Perfect. Yes.
Okay. Well, thanks, everyone. It's been really interesting talks, and I know the last ones will be too. I just wanted to say again that I'm excited about Tableau because I had tried to dabble in it, and it felt overwhelming. So I'm happy to be gently nudged in that direction. My name is Kerry. I'm here with Jason Toft, and probably some other of our lab members. We're with the Wetland Ecosystem team at the School of Aquatic and Fishery Sciences at the University of Washington. Our work is all lab-based, so my field photos will be of people looking at microscopes. But what we do in our group is we process the Neuston samples that are taken, both open water and emergent vegetation. We process the benthic cores and then also the Chinook diets. The stomachs come to us and we process those diets. So the focus of my talk will be primarily on the 2021 Neuston sampling and also some preliminary analyses that I have begun for a potential manuscript. So 2020 Neuston samples are done. Benthic cores and diets, we're still working through the data. In our lab, real quick, how we process samples is for benthic and Neuston samples, the number of individuals are counted and they're ID'd to order or family, and then they're weighed to the nearest 10,000th of a gram. And for diets, the stomachs are preserved in the field, sent to us, and then we count the taxa, identify them to genus or species, and then weigh them. So I thought I would give a brief summary of benthic data because I mostly will not focus on benthic data today. So the trends have remained pretty stable throughout the years that in that benthic core samples are dominated by oligochaete worms, and the large body size of bivalves, gastropods, and isopods also contribute to the samples. So there are three top salmon prey, which Tawnya briefly mentioned. So there's amphipods, dipterans, and cladocerans. These are the top prey. And so in the benthic plots on the bottom right, and you'll see these in other plots, amphipods are turquoise and dipterans are bright green. And so they do slightly contribute to the benthic cores, both in count and in weight. As we've heard, amphipods are further south and dipterans are in sight in nearly all years. So here's a few photos for you all. I'm going to have to up my game for next year and have people dress in Halloween costumes and do memes and stuff. I really loved everyone's photos. On the left is a benthic core that was being processed last week, an oligocete worm. Top right, some muddy benthic core samples on the shelf. And these are photos that our lab has taken or drawn. On the two on the right, top right is chironomid, top salmon prey. Bottom right is an amphipod that our postdoc, she did this drawing. And then this is the lab doing work. Bottom center is a benthic core. Bottom left is actually an AIMER, 2022 AIMER sample. Top left is a calanoid copepod. So briefly, for Neuston data, and this does incorporate 2021 data, there were no really new trends to report. Interannual trends, numerically, the dominant taxa are collembolans, cladocerans, and cyclicoid copepods. And that seemed to follow the trend with 2021 as well. As I said before, the top salmon prey of amphipods, dipterans, and cladocerans are present in the Neuston samples, especially cladocerans on the right-hand side. You can see in the open water toes that they make up a pretty good portion of the counts. Weight-wise, gastropods, hemiptera, and dipterans, a salmon prey, heavily contribute to the Neuston toes. And as you can see, amphipods and dipterans have more of a weight presence in the Neuston toes than numeric presence in general. And this is an NMDS plot, just to reiterate what I showed before within the Neuston toes. And real quick, what an NMDS plot does is it takes data from multiple dimensions and pares it down to a few dimensions just so it can be easily visualized and interpreted. And what this plot really shows is that there are three groups, really mostly two, cladocerans and cyclopoids, that are driving the differences between sites for Neuston toes. And this is average densities, so counts per net sets. And that is among sites and monthly and annually. So the second half of my talk is going to be on preliminary analyses I have started, having to do with trends in juvenile Chinook diets. On the left are three model/data groups, data for the top one. And then on the right are statistical tools used to analyze them. So what I looked at on the left is the index of relative importance, and that is basically how important a prey item is in a salmon diet. So this is in their diet. And I use NMDS in analysis of similarities to look at IRI. And the next one is energy ration, which is really just energy consumption for the fish. And then the bottom one is maintenance metabolism, which is metabolic upkeep for fish. So it's basically their cost for metabolic upkeep. And for these two, I used gamma distributed generalized linear mixed models. And really what it is the gamma distribution is used for continuous view data. And then mixed models use fixed effects and random effects. For example, a random effect would be year. So a random effect of year would account for interannual variation. So first with IRI, the index of relative importance, what it does is it looks at the frequency of occurrence of a prey item, and then its count, percent count, and percent weight in the diet. And so here's where we come up with that the three top prey for percent IRI are amphipods, dipterans, and cladocerans. Something off screen. And as many of you probably know, diet-wise, there is an upstream trend that starts with dipterans and other wetland insects heading down to Campbell Slough, bringing in cladocerans, getting to the lower reach sites, and adding in amphipods till when you get to Ilwaco, it's primarily amphipods. And the bottom three are more tidally inundated than the upper two. Since we started identifying dipterans more to life stage, we found that they are mostly made up in the diet of chironomid larvae. And then the amphipods are mostly a more Americorophium species. The plots on the right are order ellipse plots. And what it does, what these show is that through the years, and red, it's kind of a rainbow. I did a rainbow. So red is oldest, purple is the most recent year. What we can see is diets are maybe slowly transitioning or changing, but there's no years that are real anomalous. Same with length bin. For fish, that would be 30 to 59 millimeter fork length, 60 to 79, and then 80 to 99. Diets are changing a whole lot within those length bin categories. And then by site, these order ellipse circles are just reflecting what the NMDS plot shows for sites. And you can see that Campbell's Slough and Franz, the diets really do differ, which we knew that Campbell's Slough had a lot more cladocerans and Franz have a lot more dipterans and wetland insects. Sorry, my screen is having trouble transitioning. Okay. Next is energy ration, which is like energy consumption for the fish. So first I'll just explain this plot. On the x-axis is length, and on the left is energy ration. And energy ration does incorporate fish weight. So you would expect as a fish gets bigger, they have to add more energy to their diet to maintain the same energy consumption. These fish are sorted by marked and unmarked status. So marked are hatchery and unmarked are more natural fish. And larger fish in general are hatchery fish. And so you can see that there aren't any alarming trends, that really energy ration has been pretty stable. The model results that you see at the bottom showed that the highest energy ration values were actually in July and August. There seemed to be a trend of February to May, pretty good energy ration values. There was a dip in June, and then July and August, they increased again. The highest energy ration values were also for the smallest fork length fish, which does make sense given that fish weight is accounted for in the equation. Because most fish, juvenile Chinook, were found at Welch, Whites, and Campbell Slough, the middle sites, I did some modeling just to compare among those sites, and found that the energy ration values, like what fish were getting in their diets, were similar among those sites. When I incorporated all five, Ilwaco was the highest, but they have, Ilwaco also has the lowest density. So I'm a little suspect of that. And this is not an analysis, this is just a plot I wanted to show. On the x-axis is years, and on the y-axis is energy ration, and they're color sorted by length of fish. And I guess what I wanted to just point out is that there aren't any alarming trends that through the years energy ration is decreasing. There seems to be some stability. And last, maintenance metabolism looks at metabolic upkeep. As you can see, there's some coefficients in there. Two of them relate to weight and water temperature. So as water temperature increases, as we heard, and as weight and length increase, metabolic upkeep is going to increase as well. So this plot has length on the x-axis and maintenance metabolism on the y-axis. They are color sorted by marked or unmarked status, and obviously as they get bigger, they have higher metabolic upkeep. I thought there were two interesting trends that I would like to look into more. The top left plot and the bottom center plot have diverging lines and a mixture of marked and unmarked fish. So I'd like to look into what years they account for and if there's some trend, a newer trend, because all the years are pooled in this plot. So I would like to see if there's a trend where warmer years are more recent and therefore they're facing higher metabolic costs. I also noticed in Campbell's Slough, it looks like there might be a slight divergence with marked and unmarked fish. And I'm not sure if that has something to do with hatchery release timing, releasing in better temperature regimes. I'm not sure about that. But model results show that maintenance metabolism increases with fish length and increases as the season progresses due to high temps. But I just wanted to note that the highest interannual and all season densities of fish are at these 3 middle sites, even though temperatures, like we heard, are exceeding critical fitness thresholds. And then just one more plot just to show, I've plotted maintenance metabolism with energy rations, since we can look at both when we have the salmon diet. And there isn't really a sharp decrease as maintenance metabolism increases, but as you can see, smallest fish, the smallest fork length fish, have the lowest maintenance metabolism and in general higher energy ration values. So at some point it may become advantageous for fish when they're growing and not able to maintain their energy ration values to leave the system. So in summary, the sampling effort has been mostly consistent among sites. There are fewer fish at Ilwaco and Franz, but we heard about the issues that happen at Franz for sampling that hinder sampling. Yet they may not be prey limited at those sites. It's other issues. For example, the fish at Ilwaco had the fullest stomachs, and we know that there are energy dense insects at Franz. Once again, the highest densities were at the middle reach sites, even though the temps very often surpass the fitness thresholds for juvenile salmon. And I just thought this was interesting to note. I got this from one of the reports that these off-channel sites have higher fish diversity, some species that are tolerant of warm temps and low DO, dissolved oxygen, and 6 of the most common salmon, juvenile salmon predators are found frequently at these sites. So in summary, these diet trends is that there are high energy ration values, and they're pretty steady from February to May. They decrease in June, and then they increase again in July and August. Prey and foraging behaviors may offset some of these higher metabolic costs. And salmon densities peak in May, and then they quickly decrease for small salmon, and they slowly decrease for larger, which makes me think that some small salmon are leaving the system or they're entering the next class size. And if they do leave, there's reduced competition for resources. So the title of our talk was Exploring Relationships Affecting Site Productivity of Fish Prey Quality versus Quantity. And so I'm excited about Tableau. I'm excited about working collaboratively with you all, just to go more in depth about exploring the relationships, all the different biotic and abiotic factors that go into productivity for salmon at these sites. I wrote a few examples like bioenergetics modeling or pairing diet data with stable isotopes and biophysical indicators of sites. So that's all for me. Thanks.
Great, thanks. Any questions for Kerry? Oh, Curtis.
Yeah, really interesting. I've often wondered about the importance of the various prey types to salmon diets, and you've got your IRA, which is just a measure of abundance and the weight, and you've got other metrics that use the energy, of the different things, but the energy content of the prey seems to me the important parameter. It's the weight times the energy density. So for each individual, say, cladoceran, I think they're like mostly water, aren't they? So, there's not as much energy, something like that, than as an dipteran. So, in a way of thinking of what they're eating, when I think about them eating cladocerans, it's like you're just eating like water gumdrops. There's not much there versus like one, beetle or something, which it's just, stock full of energy. So I wonder if we should start considering, you know, the importance of the energy content of the prey rather than some of these other metrics.
Yeah, that's really interesting. I have wondered like how opportunistic they are as feeders. And when you're talking about energy content of the prey, though, that is incorporated into the equations. Like, yeah.
Okay.
Oh, does that
Yeah,
answer your question, or did you?
Okay, so the energy content, so energy density is just the amount of energy per, you know, weight of the creature. But then when you think about each creature, it weighs something, right? So when you factor it through, you have that creature has this much energy associated with it, right? So that's what when I look at all the different prey items, to me it's like, which one has the most energy? Because they're eating a variety of things, bigger, smaller, more energy dense, less energy dense.
Yeah, I'm sorry if I'm not understanding the question right, because energy rations do incorporate the energy, like the value of that energy, which is known like for a whole bunch of invertebrates, into that equation. Jason, can you answer that better if I'm not understanding the question?
No, I think you're understanding. I mean, I think Curtis's point too is, I mean, really what you're talking about is taking the next step to a bioenergetics model. Which,
Oh, yeah.
You know, which we've talked about, we can do, the data's all there. We just haven't had the time to piece all this together. And so the energy ration was kind of that first step of like looking at that. Yeah, and maybe there's a way, given your question, we can better express that next when we write it up on just how, you know, just the energy content of one cladoceran, say, versus like how many are in their diets and making sure we're rolling that up appropriately.
Oh, yes, I get it. Thank you. Yeah.
Yeah, that's what I was getting at. And you know, just as a conceptual idea, okay, there's all these different prey items out there. How many cladocerans do you have to eat to equal one beetle? It's a lot. So that's just kind of what I was getting at. And you're right, they're eating all these things. They're definitely consuming almost opportunistically things that come by. And right, but some things are worth more, you'd think, than others, and perhaps with less foraging, you know, energy wasted with getting all these little cladocerans, you can eat one beetle and you got the same meal.
Yeah.
That's all I was getting at.
Okay.
Didn't Mary Ramirez do that a few years ago with Jeff Cordell? I think they did.
Yeah.
They had that a whole
Yeah.
science work group presentation on exactly that. Yeah, they had the table with all the different. Yeah.
Okay. Well, thank you. I'll try to do that next year because it's there. I'll add it in.
And I will say that it is. Yeah, it's been historically part of the analysis, and I do think that Kerry and the group have continued that work. It's just the way that she presented it was slightly different. But Curtis, if you go to the EMP report as it stands, that analysis is in there as well.
Okay, sorry about that then.
No, it's good for us all to be on the same page.
Any other questions for Kerry, or we are going to move over to fish? All right, the last but not least presentation of the day. I think Jeff, you're on the hook, right? Jeff and Susan, and I think Susan is participating as well. And there it goes. And Curtis gets to take the back seat today because this is his first science work group in a while. So maybe next year Curtis gets to take more of an active role in the presentation. We'll put you on the hook next year, Curtis.
We'll see about that.
Can everybody see that?
Yeah, looks great. Thanks.
Okay, great. Well, I'm glad to be here today with everybody. Can everybody hear me okay? It's a little spotty earlier, so just making sure.
No, you sound great. Thanks, Jeff.
Okay, great. So like Catherine said, or actually what Sarah mentioned earlier, that Reagan was the lead of the fish side here at NOAA for this project for, I believe, since 2016, 2017, when we took it over from
Lyndall Johnson.
What's that?
Lyndall Johnson and the
yeah,
group
with Lyndall Johnson, Sean Soul, and Dan Lomax.
Oh, okay.
And we took that over, Reagan, Susan and I took that over the sampling portion over in 2017. And now Reagan left this year in July, and we were lucky enough to get Curtis to come on as the new lead for us. And we were just about finishing, or we did finish up our sampling in June, right when he came on. So that's kind of why I'm chosen to do the presentation today. So let's see, moving on. Okay, today I'm going to just give you a little brief update of what I'm going to discuss. I'm going to first talk about the sites. I won't talk about it too long. Everybody else has kind of talked about the sites and where they're located and everything. So we'll breeze through that. And the physical properties, specifically temperature and DO. We take temperature and DO at the site for each time we go and sample. Not nearly as in depth as Joe's information. So it's just that's just kind of a snapshot for the day, the temperature and DO for the day that we actually sampled. Fish community. This project targets, we specifically target Chinook salmon and other salmonids. However, we catch a number of native and non-native fish in the lower Columbia River estuary. And so we'll kind of explore that and see what other fish we catch at these sites and the dynamics that are at each site. Of course, the salmonid community, we'll look at that, and as well as looking at our pit tag construction. Traditionally, we've ran a pit tag array at a number of sites. Now we're actually currently just focusing in on one at Campbell Slough, and that's actually currently being restored or reconstructed, and we're in the middle of that right now. So we'll kind of, I'll give you a little snapshot on that at the end. So. But real briefly, we have the 5 sites and the 5 different reaches. Unfortunately, this year when we went, we usually start our sample in February, and this year we had that little winter bout, I should say, of COVID, and because of some NOAA protocols, we were unable to actually get out in the field in February. We're not actually able to be at the office either. So, but as soon as that lifted, I believe it lifted like the 12th of March, and we were, I think, out sampling that next day or shortly after. Are the slides advancing okay?
I'm still seeing the first one, but I think I'm seeing you in like your document recovery sort of thing. So no, I'm not. I'm just seeing the very first page.
Interesting.
Yeah.
If you have multiple screens, you might be sharing the wrong one, Jeff.
Yeah. Well, maybe if I close, you won't be able to see me, but maybe if I close this, does that help at all? Or
It's still showing the first one.
still showing the first slide, huh?
Yeah.
But it's interesting.
Maybe try the slideshow, running the slideshow. Right now we're just seeing the PowerPoint slide with all the information and not the slideshow.
Yeah.
Yep.
That's
well,
good
I
idea.
have it on the slideshow on mine. Still not working?
No.
Let's see here.
Well, now
that changed it.
Yeah, now it's on
Okay.
3.
Oh, I took it actually out of slideshow there. So
You're still not, it's kind of weird. We can see the screens, but.
Jeff, do you see the document recovery panel? If you close that, that might help us see your presentation better.
Document recovery panel?
Yes. It's right next to your slideshow.
At least according to what we're seeing.
Maybe try unsharing and then resharing your screen. That might help.
Okay. Now I can't find it anywhere.
Well, that didn't work. Sorry.
That didn't work?
No, it's actually much smaller. It's odd. Yeah.
This was an issue yesterday for us too, so.
Oh, interesting.
Jeff, if you want, you can email me your presentation and I can do it. Oh, wait, this looks like it's working.
Yeah. Looks like
Is
it's
it changing
better.
slides or?
Yeah, it says it's on 3 now. The map.
Okay, is it changing to the temperature or no?
No.
Not yet.
Gosh, dang it.
I think you need to go to slideshow and then go from current slide.
Yeah, that's exactly what I'm doing.
Oh,
So right now, I've tried a few different things. How about I just send it to you, Sarah?
Perfect. Let's do it.
Okay. Hopefully it'll send it quite large.
Catherine, maybe the group wants to do some discussion while we sort out these technical issues.
Let's see.
Um,
I'm afraid it's too large to send.
yeah. It probably could be.
Let's
You've
see.
only got
I
14
could-
slides?
What's that?
It says you've got about 14 slides. Does that sound
Yes.
right?
Yep.
Okay. Do you,
I could go
I mean,
down
we
and
could-
try it on Susan's computer with Susan
I think we could
downstairs.
see it.
You could-
Okay.
Save it as a PDF and send it to me that way.
Yeah, there you go.
I don't know why this is not working. I'm doing everything.
So we could have the conversation about if anybody has any questions that we did not answer with any of the previous speakers. We could talk about that for a little bit and while we try to figure this out. Does anybody have any questions for any of the previous speakers? Sorry, Joe. I'm just looking. Yeah, I'm sorry. I just was looking at your slideshow. It's kind of interesting. It's kind of like taking up most of your slide is like too big or something.
Yeah, it's like the screen resolution doesn't allow us to see your entire slide.
No, that's a good way to describe it.
That's better.
Yeah, do you want to try that and
Try to advance the slide, Jeff.
Yep, hold on 1 second. I just tried a few things here.
bunch of chefs in the kitchen, Jeff?
Yeah, this is a concern. Now I can't even find the. Is it anything now or?
Yeah, we're getting a big picture of the first page, but it's again like the resolution is it's like the slide is too big for the screen or something.
Shoot.
Yeah, it's not quite working. Is it possible to save it into PDF and try that? Oh, it's advancing though.
Yeah.
But can you convert it into PDF and try showing it that way?
Sure.
So again, does anybody have any questions for any of the previous speakers? You could probably have a couple minutes to discuss some of the presentations earlier. So there is some discussion, I think, Sarah and Ian touched on this about like converting our annual reports into some kind of hybrid format where some of it's in Tableau and then some of it's still in the so-called hard copy, if you will, electronic format. I think a lot of people don't or have not been reading our annual reports. So we're hoping that this will make it a more user-friendly way of showing the data and disseminating information. Is there any questions about that so far?
Yeah, hi, Catherine.
Lynn,
Catherine,
yes.
yeah. My only question would be, maybe you've thought about this, is that can Tableau preserve, you know, we have to keep records of implementation if it's related to the buyout. And so can Tableau archive all that information so it's accessible and or provide it in the background in a way that can be uploaded to CB Fish? Those would be my questions. Thanks.
I just want to jump in here and say that I was actually having a hard time picking up some of your audio. Could you repeat your question? I'm sorry about that.
I'll lean toward my laptop. So, you know, with NIMS, and if it's related to the buyout, we have to have a way to archive implementation records. And so does Tableau. It doesn't have to be in a big narrative report, but, you know, does Tableau have a way to ship, you know, some format by which all that information can be available to us as a long-term record of implementation and or shipped to CB Fish for access?
Yeah, that's a good question. And so I think that there are a couple different layers here. One is that depending on exactly how we roll Tableau out with the EMP researchers, you know, they will have that historic data set in Tableau that can be archived in the data itself, depending on the, you know, the format that you would want. And then on top of that, the dashboard interactive platform, that's going to have pages like a website pages. And each of those can be exported as a PDF or as a PowerPoint slide. So those are all easily archived to take like a screen, like a capture in time. And that's something that would be sort of part of that report out. You know, we could PDF all those pages that are created for the EMP report update. But again, those are, you know, different types of archiving of that data and analysis. You know, there's the database archive that we would be, you know, would be updating the databases across the different EMP researchers and then having that archive. And then taking, you know, essentially creating a PDF document of all the dashboards that are created. Does that answer your question?
Yeah, that's helpful. And just so as you're thinking about it, as you make your plans, we wouldn't need every temperature measurement, for example. It would just be at the same level of what you might have put in a narrative report, you know, or the appendices to that report. It wouldn't necessarily be every single data table. It's a record of the synthesis of what you learned that we could then have as an implementation record for the buyout.
Okay, yeah, and I think that, you know, what would be really helpful, Lynn, is if, and we can send this to you directly, if you take a look at the hybrid AIMER report to see if it fulfills your requirements or your needs. And so we have the you know, essentially the PDF report, executive summary and background document, and then supporting that are those Tableau dashboards. Those Tableau dashboards can be printed to PDF and created, you know, and sort of, you know, archived that way, or maybe that written PDF supporting report is enough. So it would be great feedback to get from you if you had time.
Sure, thanks.
Here's a question for the group. It's 10 till noon, and the meeting is supposed to end about noon. What we could do is see if Jeff is able to just give us some hot takes from the presentation and then see whether he can record his presentation later. Or maybe he's come up with, oh, he's got Susan showing it. But hopefully everybody can stick with us for a little bit. But again, if you do have to leave a little bit early, we are recording this. So if you have to leave, you can always we'll post it on our website so you can come back and I will send out a link to the recording so you can look at that if you end up having to leave and miss the presentation on your fish. There it sounds like Susan is with us.
Yeah, I've gone down to Susan's computer to try to work on it. Do you see anything now, or?
Yeah.
Or is it still?
No, it works, Jeff. Looks like it's working anyways.
Just move slide. Did you see that?
Possibly you had to speak too soon. No, I did not see it. It's still on the first slide. Sorry. I'm so sorry, Jeff. This is no, it's
fine. I feel bad. I'm really sorry. It's not advancing?
No. You know what we could do, Jeff, is what I was thinking is if you want to, we can help you record this later. So,
yeah.
Oh, wait, there you go. It just advanced now. It's on the second slide now. Oh, now it's on the third. So now I think it's working.
Can you see it? Can you see it or not?
Yes, we can see it.
Can you see all the graphs on this one?
Yes, it seems like it's working now. You're on temperature and DO.
Okay, so I'm gonna go. All right, so for interest in time, I won't redo what I did before. A lot of it was actually already covered, and so I'll just pick up with this year's sampling. And I want to point out that we're going to be, what I'm going to be talking on is this year's sampling, the 2022 season. We do have one more sample to do. Actually, we're doing one tomorrow and Thursday and Friday. So we do have our last October sampling to go. But for the most part, what you'll see here is pretty. October sampling we won't add a lot from the salmon side, and so what you see today is pretty good preliminary results for 2022. I also want to point out that the table shows the number of efforts sampling that we have. If we catch 30 Chinook in the first sample, in the first bag seine sample, we are done. If we don't catch 30, we continue for 3 more efforts. And if we catch, we will take genetics and keep 30 fish for odorless and lipid analysis, and some of those samples also go to the other groups, like you've already heard today. As well as if we catch up to 100, we'll measure up to 100 fish, but then count everything after that. And so moving on with that idea of our sampling regime and our sampling protocols, we also take temperature and DO at the sites. What you'll see here this year, like Joe had mentioned, was a cooler spring. It was definitely a wet spring. We definitely were under an umbrella quite often this year. But with typical trends in the temperature, as the season goes on, temperature increases and generally DO decreases. These are very typical trends. Sometimes in the month of June, the temperatures will exceed that 19 degrees that they talked about earlier, that 19, 20 degrees where it starts to be very stressful to handle the fish, so we don't. However, this year we had no issues with that. Everything was under that threshold. So we were able to sample all the sites all the way through June. So with the fish community stuff, one thing that, so I'll have this year's graphs that will show kind of what we caught this year, and then it'll also be with comparing kind of all the years and all the sites. So there's a lot there to kind of take in for the all site graph. So today I think you're just going to kind of maybe want to focus in on some of the trends. I'll try to point those out. We didn't see a lot of variability this year compared to other years. It's very similar trends. The brown, so this is the fish communities. They're all listed as family because we get a number of different fish that incorporate or that are into a certain family. So we can't really list all the different species that we catch. But I'll try to point out some of the big ones. As you see, the brown is a very predominant color for this year and for our yearly site or yearly data. And that's sticklebacks. They make up primarily or the most of any species we catch. And then you'll see the light blue, and that's the salmon. And so with Welch and white, we don't have a lot of diversity, species diversity in those sites. And so the stickleback and salmon make up predominantly the most of that. You'll see Owako, Campbell Slough, and Franz have a little more diversity. Those consist of more of, they still have that stickleback base, but they also have a lot of the Cyprinidae, which in this case is, for the most part, would be the northern pike minnow, which is a predator. Another predator that we see in our sites is also it can be a predator and a competitor at the same time, but the Persidae, which is in this case would be yellow perch for Campbell Slough. Those are the main fish that we see at these sites. We also see some centrarchids at Campbell Slough, and that consists of bluegills, crappie, and we also see some largemouth bass. That's generally a little warmer system. Campbell, it's further from the main stem. And Franz Lake, we don't get to sample that as much as we'd like. Some of the gaps that you see that you don't see a sample year for, that's generally because of flow issues. I'll point this out now because you'll see this throughout the presentation. Franz Lake, once the Bonneville Dam gauges show about 17 feet, it's very channelized in Franz Lake, and so once the water gets above that channel, it really floods into a lot of willows, and it makes it almost impossible for us to sample in there with a vague seine, even with a pole seine. And so we're unable to get a sample there a lot of the times. But as you see, this year follows very similar trends as previous years for the fish community. Moving on quickly with the seasonal and site composition of salmon, as you see that most site, Chinook is that gray color, the Chinook, and that's the predominant salmonid that we catch. Owako, we traditionally will catch more chum than any other site. And this year we saw that the most chum we saw was in March. Typically that's in April and May. And this year we also saw the most Chinook in April, and that's typically in May. So everything was a little bit a month earlier on a few of those highest densities for the species. A total of 132 chum were caught. That's the second most. We did catch a coho in Franz Lake, and we did occasionally we catch a steelhead, and this year we did catch one at White's. I'm looking into the Chinook density. As you can see, March was our highest density for unmarked Chinook. Marked Chinook was the highest also in April. However, typically we see that probably more towards the May season, but this year we saw a lot more in April. It was very good catches this year, the cold spring. As you see, for all years, it's kind of hopefully not too confusing of a graph, but because we have some we never catch a lot of unmarked Chinook, or I'm sorry, marked Chinook. So, and we do often will have high density catches of unmarked Chinook. Therefore, graphing it sometimes can be difficult. So those numbers above the bars are the actual values in which we caught those years. So you see Welch and White are definitely our strong sites for for just total abundance of Chinook salmon. And as you can see, Campbell Slough, we get a variety of marked and unmarked. It's definitely the site where we see the most marked fish traditionally year after year. And so then moving on to percentage of marked and unmarked Chinook, we kind of touched upon this a little bit, but once again, so Ecological shows that we had the most amount of marked fish, or the highest percentage of marked fish, I should say, but the most marked fish will actually came from Campbell Slough again, like it traditionally does. We only caught 6 fish at Ecological. However, we're very proud of those 6 fish because in the past we've had trouble catching fish there, which is kind of frustrating for us because that's a site where we can sample very well. We can basically cover the whole. We have a 50-meter bag seine, and we just kind of pull it across the channel, and we can cover we cover the whole section that we sample. And unfortunately, we're just unable to catch some Chinook in there year after year. So these are fairly similar trends. As you can see, Campbell Slough actually, for the amount of marked fish we caught this year, was down from most years, or from previous years. Some years we can see as high as 90% marked fish in Campbell Slough. Usually it's around that 40 to 50%. This year was closer to 20%, which is a little lower than normal. And so now this is moving on to the genetic composition of, so we're going to take a little bit of a turn here, go back. And so I was presenting on the 2022 data. Now we don't have the new genetics data for this year, of course. So, but we do have the genetics data from last year's fish, the 2021. So we're going to look at those and compare those with the previous site or previous years. And so we have unmarked on the top and marked on the bottom. And you'll see that there's some definite trends here. You see that Welch and White and even Ecological, although it's very few fish caught there, is predominant, and you also see it in Campbell, but West Cascade Falls is our predominant stock. We saw that again this year in Welch and White. And when you incorporate the marked fish into it, you also see West Cascade Falls, but you also see a significant amount of the Spring Creek group. One thing that we're potentially going to look into, and something that's kind of interesting, is that the diversity of the composition of stock that we see in Campbell and Franz, the sites that are further upstream. With the marked, but you see that in unmarked, but not so much in the marked. You see, which we would tend to think that would happen, that we'd see mostly the Spring Creek group there. So let's dive into this a tiny bit more here with the length frequency of the stock from the 2021 genetics. As you can see, like I mentioned, so this is the marked and unmarked together. Welch and White, once again, West Cascade Falls dominant, but then it gets a little more interesting when you look into the Campbell Slough and Franz. Campbell Slough is very similar to previous years. As you see, with all the sites, as the fish increase in size, you generally see more different stocks. You definitely see more diversity in stocks, or multiple stocks encompassing the sample. And in Franz, like this year, we caught pretty much all except 1 or 2, and we did catch 77 unmarked Chinook in Franz Lake this year, which is a very good catch. It's the largest catch ever in Franz Lake. Unfortunately, we're only going to sample 1 time this year because of the high water, but we did catch a lot of the upper Columbia River stock in Franz Lake, which we have caught in the past. And so I just want to point out that that's kind of the information that we've gathered this year and from 2021 with the fish data. We are in starting to work on it. Well, we've been working on for quite a while the data set and trying to figure out some of this individual based fish. We're looking at individual lengths, weights, conditions of fish to try to see if there's any indicators of how fish are doing in different years. And a few years back in 2018, Paul Chitterow published a paper with some of the EMP and other lower estuary site data that had fish growth. And our sites typically, and we do take otoliths on all of our the 30 fish at each site, at each sample. And so we have otolith data, and it does show that they are a little bit slower growth down here. And so that's one thing we kind of want to maybe look into, and perhaps, get together with the diet folks as well. But there's some interesting things that we'd like to try to tease out in the future, but there's still a lot of work to do there. And so doing a quick kind of transition to the pit array. So real quick, I know in the past, we've given a this is, I think, the first year that we actually don't have a pit tag array currently working. We decommissioned and dismantled the horsetail array in 2020, 2021. I remember last year we Reagan gave an update on that. Some of that is being used for Campbell Slough and I believe the new Staggerwall pit array. So today I'm just going to kind of real quickly focus on what's going on at the Campbell pit array. And so it's originally in 2010 it was established, the pit site, and then it was revamped in 2018 because it didn't have full coverage and it experienced a lot of heavy debris, and so it was essentially broken. And so we kind of came in and had these large PVC arrays that we hung on a line, and that worked good for a few years. However, as you can see, we encompass a lot of big trees and debris and things that can cause a complete headache when you're trying to run a pit array. And so that kind of took us out during 2020. We couldn't look at it for a while during COVID, and then when we came back, we had this giant, as you can kind of see in some of the pictures, we had that giant tree come and take us out pretty good. And we also had some vandalism that took our solar panels and batteries. So we had a bit of a rough go there for a little bit. But so we were funded we're given the opportunity to put a new array in there, and that's what we've been currently working on. It is going to be a flexible antenna style. It's actually going to be about 230 feet, which will stretch across. It'll be in a figure eight. Pardon my terrible drawing over there, but it'll be in a figure eight type style. It's about between 60 and 70 feet across the channel. As you can see, this is during very low water. This was actually just in the last week we put these marsh posts in. So we're hopefully going to be putting the array or the antenna in very shortly in the next few weeks. And so we're really excited about that. And so we think we'll get a little bit better coverage, and we're just excited to get something going there again. So I know I'm way over time, and I'll really quickly just kind of highlight that, that this was a great year for catching Chinook on the Columbia River at our sites. We got marked and unmarked at all 5 sites. The fish community patterns seem to hold pretty steadily. Once again, greatest diversity at Campbell and Franz. The typical season geographic trends of Chinook and chum, the highest densities of Chinook in April. Overall densities are similar to 2021, except El Waco, where we caught fish this year, and we're really excited about that. And whole sites dominated by unmarked Chinook, which is not uncommon. And the similar stock diversity scenes at the site this year. However, we did see a lot of, or pretty much predominantly all of the upper Columbia River summer falls at Franz this year. So that's very interesting. And here in the next few weeks, we will have hopefully be getting Campbell Slough pit tag array in, and hopefully be operational for the 2023 season. And so, and I'd also like to thank Kerry Lyons here at Point Adams for helping with the data entry and for the fish processing that we do at the end of the year every year. So with that, thank you, and take any questions if we have time. Probably, I don't know. I'm not sure after my issue here with the PowerPoint. I apologize for all the issues.
Yeah, appreciate that you guys stuck with it and were able to do it. Sorry about that, Jeff. I wish I could have helped you a little bit more. Sorry about that.
No, that's not your fault at all. That's all on us. That's on me, I should say.
So how about any questions for NOAA, for Jeff? For anybody that, oh, Kerry.
Hi, I have a quick question. Why do you think there are so few Chinook at Ilwaco when there are a lot of chum? And why do you think there are so many marked Chinook at Campbell in particular? I know that's a hard question to answer, but just wondered if you had thoughts.
Yeah. So I will address the Campbell or the Ilwaco one first. You know, I'm not really sure why they're not in, they come into the slough, into that. So we sample probably about, 20 meters into this side channel. And I know we know for a fact after that there is Chinook in that area on that point by the Mainstem, but they're not really, it seems like we're having a hard time catching them in the sampling area. I'm not sure why that is, because you know we fish it as high tide goes up, and so the slough is flooded when we get in there. So it just seems to be something about that area that we're not able to have them come into that area. It's a tough question, or it's stumped us for years, to be honest with you. But that's the only thing, unfortunately, we can kind of think of. And then the Campbell Slough, why we see more hatchery fish at Campbell Slough is. Boy, I wish I had an answer for that, but it's kind of yeah. I think the only other reason that, you know, we catch most of those come May and June, which that fish is generally a larger fish then, and so maybe that fish is just larger, has better swimming ability, and can explore areas more, because that site that we have at Campbell Slough is the furthest from the Mainstem. It's about a mile from the Mainstem, and so maybe that area has a lot of, it is a little warmer there, so maybe they're going in there, or I'm not sure why the hatchery fish would select that, maybe. And we also see, stock or a variety of stocks in there, so that also might be a reason why. But all good questions, but unfortunately I don't have as good of answers as I would like to have. But does that kind of answer? I'm sure it doesn't answer it, but that's what I got.
Yeah, thanks. I just wondered if there was something, really obvious or not. I assume there wasn't, but yeah, thank you.
Not to us, but if anybody has any insight, we'd love to hear it.
Great, thanks. Any other questions?
I was just going to say that, as we synergize the data set, some of these patterns we probably will have more questions than answers, but, it's also good to have, to help us develop hypotheses about the patterns we do see.
Yes. Completely agree.
Okay, if no other questions, it's 12:15. So thanks everybody for sticking with us. I know we went over a little bit, but so we will be posting the recordings from today and plus the presentations and on our website, and I will send out the links when we get all that information posted. But thanks everybody for sticking with us. Great presentations, and thanks to all the presenters. You guys were all awesome. Thank you so much. And again, thanks, Jeff, for sticking with it. I know that was a little stressful.
I'm sorry. I don't know what happened.